In Unit 3, Lecture 2, Part 3, we're going to look at the first of two SoftChalk lessons on how the ability to resist innate immune defenses helps bacteria to colonize the body. And in this SoftChalk lesson specifically, we're going to look at how bacteria can resist phagocytic engulfment, that is the attachment and ingestion stages of phagocytosis, as well as how they can resist antibacterial peptides. And remember, we talked about the steps in phagocytosis as well as the complement pathways and antibacterial peptides in the previous SoftChalk lesson. So once again, as always, look at your fundamental statements and bullet points and make sure you answer your detailed learning objectives and learn them for the Unit 3 exam. So again, as we've been doing for a while, we're looking at virulence factors that promote bacterial colonization of the host, the ability of bacteria to establish themselves in or on the body, one of the first steps in causing diseases. And we've already looked at the first four. So what we're going to look at in this and the next SoftChalk lesson is the ability to resist innate immune defenses, things like phagocytosis, complement pathways, antibacterial peptides. And in this SoftChalk lesson specifically, again, we're looking at the ability to resist phagocytic engulfment, that is either the attachment or the ingestion stages, as well as the ability to resist antibacterial peptides. Now, what we're talking about largely in resisting phagocytic engulfment are bacterial capsules that we learned about back in Unit 1. We learned that they allow bacteria to resist phagocytic engulfment. And of course, there's quite a few bacteria that produce capsules. Some classic ones are the pneumococcus, streptococcus pneumoniae, common cause of sinusitis, ear infection, lobar pneumonia, and meningitis. Haemophilus influenza type B that causes a variety of respiratory infections. And bacteria like Neisseria meningitidis that causes meningococcal meningitis, the anthrax bacterium, Bacillus anthracis, the whooping cough bacterium, Bordetella pertussis, to name a few. And this is a photomicrograph here of the Klebsiella aerogenes that we looked at in lab 6 in the capsule stain. So remember that if the bacteria secrete a glycocalyx that tightly adheres to the organism and they secrete that in abundance, the capsule is so named because it encapsulates, in this case, the bacillus. So the clear area there is the capsule, and of course the purple rod inside is the bacillus. And that is one of our highlighted bacterium, Haemophilus influenzae. So capsules, well, we learned back in Unit 1 that capsules resist unenhanced attachment. Capsules prevent PAMPs on the surface of the microbe from binding to endocytic pattern recognition receptors on the phagocyte. So again, if the capsule is thick enough and dense enough, it covers up the PAMPs on the surface of the bacteria, preventing the pathogen-associated molecular patterns or PAMPs on the microbe from binding to the endocytic pattern recognition receptors. And remember, PAMPs binding to endocytic PRRs is what we call unenhanced attachment. So capsules are blocking unenhanced attachment. And we showed you this animation of that, some animations of that as well. So that's one of the major roles of capsules initially, because remember, unenhanced attachment is our first attempt to stick the microbe to phagocytes so we can destroy it. But if the PAMPs are covered by the capsule of the bacterium, then they can't bind to the endocytic pattern recognition receptors. So if we can't engulf the bacterium, we can't kill it, and the organism continues to replicate and increase in number and potentially cause disease. But fortunately, we have a body defense against these capsules. Now, the capsule itself is resisting unenhanced attachment, again, covering up the PAMPs so they don't bind to endocytic PRRs. But capsules act as antigens, and we can make opsonizing antibodies against the capsule. So this shows you the role of the capsule here. Notice that the PAMPs on the surface of the bacterium are covered by the capsule. So when the endocytic PRRs try to grab a hold of the PAMP on the bacterium, it can't get a hold of them, and it can't stick the bacterium to the surface of the phagocyte, so the organism is never attached. But remember, if it can't be attached, it can't be engulfed. Now, occasionally, if it's pushed up against some solid surface, it can still take it in. But basically, the capsules are resisting this unenhanced attachment. And we see that happening. This, I believe, is the pneumococcus streptococcus pneumoniae. And because it has a capsule, it can't stick to the surface of the organism, so it just gets pushed around by the neutrophil. Now, later on in this movie, the neutrophil does eventually engulf it, but it actually pushes it against some solid surfaces. So the capsules give a bacterium a temporary advantage by resisting unenhanced attachment. Now, another way that bacteria can resist engulfment is with capsules is through a different mechanism by interfering with complement pathways. So capsules can sometimes interfere with the body's complement pathways. We'll give you a couple examples of that. There are some capsules, Staphylococcus aureus down under part D is one of these, that inhibits the formation of C3 convertase. Now, this is an enzyme in the complement pathway that breaks C3, which is inactive, into active C3b A and C3a. And of course, once you split C3, then you get all the other parts of the complement pathway too. So basically, if the capsule prevents formation of this enzyme, then the opsinin C3b and C4b needed for enhanced attachment are not made, as well as all other beneficial complement proteins like C5a that come later in the pathway. Without that early enzyme, the complement pathway is interrupted. Sometimes capsules simply cover up the C3b. If there's enough capsule there, it may prevent the capsule from making contact with C3b receptors. But again, as we said, the body has a way around this capsule. We produce eventually opsonizing antibodies against capsular antigens, against epitopes of the capsule. These are the opsonizing antibodies IgG, and then these antibodies stick the capsule to the phagocyte. So as we see in this animation, this bacterium is surrounded by a capsule, and these are epitopes sticking out of the capsule, short branching sugar chains that are part of the antigen capsule. So we make antibodies where the FAB portion of the antibody is tailor-made to fit these epitopes on this bacterial capsule. The FAB, or antigen binding fragment of the antibody, then binds to the epitopes of the capsule. And then the FC portion of the antibody binds to FC receptors on phagocytes. And in this way, we can attach encapsulated bacteria to phagocytes, engulf them, and destroy them. But the only problem is, if we've never seen that capsule before, it could take us 5 to 10 days to make protective levels of antibodies for opsonization. And during that time, the capsule is resisting unenhanced attachment. So what we do sometimes is we teach the body how to make these opsonizing antibodies against the capsule ahead of time. And we inject this capsular material as a vaccine. So we have 2 vaccines against pneumococcal pneumonia that contain capsular materials from the most common strains that infect children and the most common strains that infect adults. And so by injecting this capsular material, we teach the body how to make opsonizing antibodies against the capsule before the bacterium ever gets in the body, and develop memory cells so that if the bacterium does enter the body, we get a massive production of antibodies against the capsule very rapidly, and they immediately stick the bacterium to phagocytes. So when we immunize against pneumococcal pneumonia, we're injecting capsular material to teach the body how to make opsonizing antibodies against the capsule. When children get the Haemophilus influenza type B vaccine, or HIV, we're injecting capsular material. Some of the vaccines against meningococcal meningitis use capsular materials. And remember, of course, that many pathogenic bacteria as well as microbiota produce biofilms. And through the norm form sensing that we learned about in Unit 2, this allows bacteria to eventually adapt to their environment as a population rather than an individual bacteria. And by living in a community in a biofilm, that provides several advantages. The bacteria are more resistant to attacks by antibiotics. They can trap nutrients for growth and remain in a favorable environment. They can adhere to environmental surfaces and resist flushing. They can communicate with other bacteria in the biofilm, acting as a population. And of course, inside the biofilm, they resist phagocytosis and attacked by the body's complement proteins. And some bacteria can resist phagocytic engulfment and complement using mechanisms not involving capsules. As just a couple of examples here, Staphylococcus aureus, a common cause of accidental and postoperative wound infections that causes abscesses of the skin, produces an enzyme called coagulase. And coagulase causes fibrin clots to form around the staphylococcus, which helps it to resist phagocytosis. Remember, fibrin is one of our own proteins. It's part of our clotting system. And so when the bacterium becomes coated in fibrin, we don't recognize the bacterium as foreign. Our immune system just sees human protein, fibrin, and not the staph underneath. And that can make it more difficult for us to recognize the Staphylococcus aureus and remove it through phagocytosis. So there's a number of bacteria that coat themselves in human protein sometimes to mask themselves from our immune system. Pathogenic Yersinia, like Yersinia pestis, that causes plague, contacts phagocytes and uses a type 3 secretion system to deliver proteins into the cytoplasm of the phagocyte that depolymerizes the actin. Now remember, it's the actin microfilaments that push the membrane up and allow pseudopods to form. So by injecting molecules that depolymerize actin, pseudopods never form, and the bacteria are never engulfed by the phagocyte. So they're actually resisting the ingestion stage of engulfment. And we said that very few gram-positive have pili. Streptococcus pyogenes that causes strep throat produces pili. But in this case, the pili block activation of the complement pathways on the bacterial cell wall. And by blocking the complement pathways, of course, they can resist phagocytic engulfment by preventing the formation of C3b and C4b, the opsonins, and interfering with the complement pathway in general. So there's a number of ways that bacteria can resist phagocytic attachment, or in some cases, phagocytic ingestion. And then finally, certain bacteria can resist antibacterial peptides. As we mentioned in the previous SoftChalk lesson, we have these antibacterial peptides produced by our immune system, including human defensins, which are short peptides about 29 to 34 amino acids long, and they're directly toxic by forming pores in the cytoplasmic membrane of a variety of microorganisms, causing leakage of cellular needs. And they can also activate cells to promote inflammation. And then cathelicidins are other antibacterial proteins produced by a number of cells. And the two peptides, upon cleavage of the cathelicidin that are formed, are toxic again to a variety of microbes. So these are all antibacterial peptides that can kill a variety of microorganisms. Well, capsules can help antibacterial peptides from reaching the cytoplasmic membrane. Often these antibacterial peptides can't penetrate the capsule surrounding the bacterium, so they never get to the cytoplasmic membrane to form pores. Gram-negatives have a polysaccharide or sugar chain sticking out of their LPS. And so this LPS of the gram-negative cell wall binds these antibacterial peptides, and that again prevents them from reaching the cytoplasmic membrane. And then finally, some bacteria secrete peptidases that actually break down antibacterial peptides. So again, these antibacterial peptides are an early part of our innate immunity that are toxic for a variety of microorganisms, but bacteria can resist those antibacterial peptides through several mechanisms. And again, anything a bacterium can do to resist innate immunity is going to make it more likely that it's going to survive within the body and not be eliminated. And again, at the end is your self-quiz for that SoftChalk lesson. In the next video, we'll look at how bacteria can resist phagocytic destruction once they are engulfed.